I wanted to talk to you about some work that I did as part of my PhD thesis, which was the forerunner to some work and some a company that many of you may be familiar with that was developed later, a company called PowderJect. You'll be hearing a little bit more about PowderJect in one of the talks afterwards. But what I really wanted to talk to you about was the work that led up to that. So part of my PhD thesis, and I've kind of jokingly called it From Star Trek Fantasy to Clinical Reality. And the joke is that, of course, every time we ever talk to people about this, once we started talking about a so-called drug gun, people would say, oh, you mean like in Star Trek? And of course, many people were Star Trekkies, but I was not one of them. So I had no idea what they were talking about when they started to talk about this. So, of course, I didn't have Google at the time, but I did finally have a chance at some point to go back and see an episode or so, and I got a sense of what it was that they were talking about. But so what I want to do is talk to you a little about the technology and some of the work that led up to the start of PowderJect. And then at the end, I'll talk to you a little about some of the stuff that I've been doing lately that hopefully has been inspired, that has been in fact inspired by Brian and his ethos and his very important effects that he certainly left to me. You've seen this picture already several times, the MEU, and of course, it's still there and it hasn't changed a bit. It's certainly worth a lot of great work went on down there over a number of years. And one of the things I really wanted to emphasize, it's been emphasized already during this talk, and it should be emphasized again and again, was the tremendous work that these three guys did, and the forerunners to them, for example, Francis as well, with the three technicians that I was very fortunate to have worked with over the years. Those guys were the ones, together with the PhD students and together with the inspiration that Brian provided, those guys were essential to making all this happen. So we were very fortunate. And I don't know if the IBME is fortunate still to have technicians like this, but it's so important, certainly in our day, to have guys like this who could make anything and could take a really pretty poor drawing and turn it into something real. So I'm very fortunate. We're very fortunate to have benefited from that. So when I started, and again, when Lionel kind of started talking about this and suggested that I do a talk, I did something I haven't done in 25 years, and that's to go and find my thesis, which had been forlorn and gathering dust on one of my bookshelves, and I pulled it out, and I went to review it, and actually then I went and looked on Google and found actually it's online. Not only that, it's actually been cited 3 times. I was like, what? This is amazing. I only realized later that, in fact, it was myself that had cited it all 3 times. So it's not quite as impactful maybe as I'd hoped. But so the work that I was really doing was really to do with developing a new way of effectively transforming plant cells. And what the technology that I'll tell you a little about was using some gas dynamics. A little bit of my background, I had worked in an aerospace industry in California for a couple of years and got on an airplane and left a beautiful sunny California. I mean, gorgeous. Got on an airplane and got off on the coldest, rainiest October morning you could ever imagine at Gatwick. And I said, what have I done? But I was there, and I was there because this man had invited me and offered me the opportunity. So that was, it turned out fantastic in the end. I did question myself once or twice, especially when the winters came and the flu and various other diseases overtook me. But definitely, it's been very valuable. So a little bit of context here. What happened in the late 80s? I think it was about '89. We were approached by a gentleman named Ron Coffey from ICI, who had been doing some work and looking at some work ongoing in the States. Ron worked at ICI, which is no longer in existence now, but it was a chemical company, and they did a lot of things. But one of the things they were interested in were making new types of plants and plant seeds. And one of the things that was big at the time was how do you biologically transform, genetically transform these seeds to make them more efficient, to give them better genetic properties that you might want. And a guy over at Cornell, John Sanford, had developed a really interesting but crude instrument. It's basically a little gun using like a .22 cartridge that would shoot these particles. The microparticles were at the tip of this little kind of bullet, macro projectile they called it. You shoot the .22 caliber thing, it would fire down, and the target cells were there, and they would receive these microparticles, which had been coded in DNA, and then they would be transformed. And it was kind of a cool instrument. They called it a gene gun, but it had some control issues, and you couldn't really control it very well. So they came to us and said, "Is there a way you can develop some new technologies that might enable you to make it more controllable?" And so I think when Brian was thinking about it, he had done a lot of work in gas dynamics earlier on in his career, so he knew a fair bit about it, obviously. And he started talking about that. He brought me in and knew my background from the gas dynamics work that I had done in the States, and we started talking about different concepts of what we could do. And the idea was fairly simple. The early conceptual idea was very, very simple. You kind of just use high pressure high pressure bottle, force it through a nozzle, and get particles going at very high velocities into a target plate. And so there were a number of issues and a number of questions that naturally would come up in something like that. When you're starting absolutely from scratch, had no idea even what kind of velocities you would need, what kind of particles you would need. So you're starting basically from scratch. What kind of nozzle design, what kind of materials you're going to use for that diaphragm. So these are the known unknowns. And there are tons of unknown unknowns that we would meet, and different challenges that we would have to address as you move forward. And that is what a PhD thesis is all about. It's about not just what you know at the beginning that you don't know, it's the things you don't even know that you don't know. And that being able to address those is what makes a PhD thesis. So we started talking about doing some early stage literature work and looking at what Stanford and these other guys had done, and they'd kind of said, oh, you need velocities of about 450 meters a second. So that's kind of what we were targeting. So we needed to figure out, well, how are we going to do that? How are we going to get particles going that fast? How do you design? So we did a lot of work thinking about, this is actually the original design that I sketched out, and for whatever reason, put in my thesis. I have no idea why, but it appears in my thesis, so I was able to reproduce it here. And we used this technique called the method of characteristics, which is a very well-known gas dynamics technique. You look up any of the old textbooks, and it will explain very, very, very well how to do this. And you can nowadays, of course, you don't need to do it geometrically. Like you just say, "Hey Siri, can you design me a Mach 2 nozzle?" And it magically appears. But of course, back then we did not have the internet. The internet was still just a gleam in Al Gore's eye at this point in time. So we had to do things by hand, and it was. I remember actually a cold, windy day in early January 1990. I was doing it by hand, the geometry of this, to actually get the design of this first nozzle that we wanted to create. So we kind of did that, and we after a number of different device generations, we got to something that looked a bit like this. And you can see actually, this is a device again built entirely by John, who again I wish he could have been here too, because I would love again to say thanks to him for all the work that he did. Over the many years we worked together, but it wasn't exactly the most elegant design, and that's my fault because I'm not an elegant designer. But you can see it did the trick, and the idea was that you'd have a gas pressure, a cylinder with gas pressure here. In the beginning, we were using air. Eventually, we started using other gases as well. And your plant cells would go here, and you would basically pop a diaphragm right there, and the cells would be basically accelerated to very high velocities very quickly, and to velocities sufficient to affect a biological transformation. So again, this is what it looked like. You can see, and many people said that it was designed like a tank, and it was. It was really, really heavy, and would have done well in battlefields. But it actually sounded like a bazooka too, and this thing was loud. So there were a lot of aspects to it that probably needed a little bit of sharpening up, and that's what we did toward the end. And I will tell you a little about that at the end. But just to take you through a few more of some of the geometry, the things that we had to do in terms of understanding the flow out of this. One of the first things we wanted to understand is this supersonic flow, because that's what we wanted, but we had to verify that that's actually what was happening. So I did first a little bit of theoretical calculations on what a flow field, a jet boundary might look like coming out of a nozzle like this. And then we started to do some actual some imaging of these gas dynamics. And I was, again, fortunate in so many ways for having the various resources at Oxford. This work was all done using, with guys over at OSNI, which is one of the engineering parts of the Oxford engineering department. But they're very, very kindly offered me to allow me to use some of their flow visualization techniques. And this is a technique called Schlieren system. I won't go into great detail, but it's a very standard way of looking at a supersonic flow coming out of nozzles. And so we did, we first, this was kind of what we expected it was going to look like with, you can see a very periodic nature of the structure, and that's in fact what exactly we did see when we were able to measure this and image it over, and these were high-speed camera shots. And so you can see the supersonic structure, you can see the shock waves, the Mach disk, and so forth. And that gave us some pretty substantial confidence that actually we had at least something supersonic that was what we were looking to achieve. So that was great. We also were able to then put this Schlieren imaging kind of results together with some pitot tubes. Again, this is all very standard gas dynamics type stuff, but it was comfortable and comforting to see that actually the cylindrical the cyclical nature of the structure of these shock waves could be verified not only through imaging but also through measuring the pressure downstream from the nozzle itself. And you could do that at various time points, and at different distances. So the imaging and the visualization tied in well with the pressure measurements, so that was comforting. We also were able to, for example, go back and look at that Mach disk I mentioned. So that the Mach disk there, which is right at the first place where the shock waves come together, and you can see that what theoretically and what other researchers working in other types of supersonic nozzles had found, our data was actually was corresponding well with what they would have expected and predicted. So that gave us, again, strong comfort that what we were measuring was what we thought we were expecting. So that was important. The next thing we wanted to be able to do was to try to measure the velocities of the particles, because again, remember, we wanted 450 meters per second is what we wanted to try to achieve. At least that's what our research had told we needed. So we had the idea, and this is one of the things I wanted to really talk about from the MEU, and one of the benefits. Lionel mentioned the Tuesday morning meetings, 9:15 every morning. He had to have a graph at 9:15 and be ready. But I can tell you what, more things were kind of discussed, and more ideas shared, and more solutions offered. Some of them worked, some of them didn't, but you had that collaboration, the ability to talk and discuss, and to say, well, I have a problem with this, and someone else say, well, have you tried this? I'll tell you another example in just a second, but this was suggested by one of the guys in the lab, to putting two LEDs and two photodiodes in parallel, and basically, I won't go into great detail, but it enables you to look at the velocity, the time it takes for the particles to break that beam, and then that beam, you can measure. You can measure that very carefully and fairly accurately. And by using again, probably the same IBM PC that Lionel had talked about, we're still using even a few years later. And you could then do something where you take the two signals, and this was actually very much, I remember very distinctly, Lionel suggested using something called the cross-correlation technique, which is how you look at two signals, and this is very much an electrical engineering concept that all electrical engineers will be familiar with. But me being a mechanical engineer, I'd kind of vaguely heard about it. But Lionel very strongly, I remember this, suggested doing this. And by cross-correlating, you just slide one signal to the left against the corresponding initial signal, and the peak point, it will give you effectively the time lag, the time difference between those two signals. And I did this hundreds of times with, and had programs, computer programs to make it all work and everything. But basically, you're able, just looking at that peak tells you exactly the time difference between those two points. And by doing that, you can then work out the velocity of those particles. And two things from this slide that are important. First, you can see the difference between using air and using helium. And helium became very important later as we started working into powder jet because we needed higher velocities, and having the ability to use different pressures and different gases was very, very important. And you remember I said we needed 450 meters a second. Well, that's what we were able to show that we were able to achieve using air alone in our system. So from all of that, we were able to, and I did a lot of work in conjunction with people down at ICI in Berkshire, and very kindly, they donated a lot of time and effort to work with me, who's a complete non-biologist, I can absolutely assure you. But they helped me to get just a huge amount of data to demonstrate that, yes, you could take these tungsten particles that are coated in DNA and fire them fast enough and to the velocities needed to actually get the genetic transformation that you're looking for. And there are a number of examples in my thesis that pointed to that, and that was all very comforting and great. And we filed a patent, which was useful, but we decided in the end that this was not going to be a commercial value in terms of using it for genetic transformation of plants, because there were other things already on the market that were more portable and more handheld. This thing was not handheld, sadly. However, we thought, and based on some discussions that we were having, let's look for other applications of this kind of technology. How else are we going to be able to use this? And it struck us that maybe you could use this to deliver medications into the skin. And Charles Potter is going to talk in a few minutes about more details about powder jet once the company itself got started. But the concept was fairly straightforward, but actually there was quite a bit of cleverness in making all this work. The first thing was we actually had to figure out first off, what kind of drugs are we going to use to begin with? What are we going to use to trial this thing out? What kind of, how are you going to actually get it into a little so-called cassette so that you can reuse this and retest this over and over? And how are you going to fill this thing up on a regular basis? Again, this, for example, this is a very clever design that really John Greenford came up with, the ability to backfill this and to reuse it on a regular basis. We redesigned the nozzle to actually make it more suitable for drugs, because you wanted to hit a larger surface area. But basically, there are just a whole range of different things we had to do to take this huge device, literally it was about this high, and to shrink it down to something that would fit into your hand. And there was a lot of work and a lot of effort that went into that, and it was inspiration from Brian, and it was dedication from John Greenford, and his cleverness and his ability to create some amazing stuff. So we kind of started to think about this idea, but one of the questions we had to answer early on was, does this thing hurt? Because if it hurts, why bother? No one's going to want it anyway, right? So, and everyone, there's a lot of folklore about this, but this is absolutely true because I was there. Brian said, we got to test this out. We got to see, did this thing hurt? So I said, all right, well, what do you want to shoot? We talked about it a little bit, and we said, well, just take some salt, let's grind it up a little bit, and let's try that. And this is absolutely true, and we did it. And I said, well, I'm not trying it first. You stick your arm in. And he of course he did. And I tested it. I mean, I ran the system, did the powder ejection, and it went off. It sounded loud, but Brian came up and said, didn't feel a thing, didn't hurt a bit. Puff of air. and which is actually is the case. Unfortunately, and I then did mine, a couple minutes later. Yeah, absolutely the same. It was just a little bit of a flick. It was no pain at all. So great. About 20 minutes later, the salt, having now entered the top layers of the skin, started to itch and bleed. And for several days afterwards, I was itching that thing. But I can tell you what, we knew that it was injected. We knew that it was working. And so that was the first, thing to actually tell us, hey, this thing is actually working. Anyway, I won't bore you with too much detail about this. It's fantastic period of time in terms of, and it was a team effort of a bunch of people coming together, and we met Paul, of course, around this time, and all of the enthusiasm about how we can take this and set up a business to actually do something really real and something that's going to benefit people. That was just tremendous excitement. And again, so you can see the handheld system that we ended up with a pre-filled helium gas canister. So these became disposable ones that you could just reuse once. One of the key things was the silencer, actually, because as I mentioned, there was a bit of a sound. And we actually like to say that it sounded a bit like a nice bottle of Gros Hermitage being opened, but actually it was a bit louder, maybe more like a bit of Dom Perignon 65 or something. It was quite loud, but the silencer did improve. We did improve that over the years, and I'm sure that Charles will be able to tell you a little more about that. So was it science fact or was it science fiction? I mean, the hypo spray that was kind of, although it was not an inspiration for, as far as I know, for either Brian, certainly not for me, but it certainly was something that really we were able to get a lot of people to grab onto in terms of an idea. And I think that was one of the key things was everyone has injections and many people hate injections. To be able to have something that actually makes those passé is actually a wonderful thing, a wonderful concept. So I'm pleased to say that it has been used. It was FDA approved in 2007. It was launched by, as far as I know, by a company called Marathon Pharmaceuticals in the States. And last I saw, it was being used in a number of hospitals in the Boston area. And this was to deliver a local anesthetic called lidocaine, and particularly for pediatric use, for venipuncture and cannulations and so forth. So we're very pleased that it did lead to some benefit. I came across this picture from, I just can't believe this picture, but anyway, this is a picture taken when they were just starting to think about creating the Oxford Science Park, which, as you know, is in just south of Oxford. And Brian was one of the actual inspirations for actually having that science park built. He suggested it to the president of Magdalen, and they started thinking about it and talking about it. And so I remember they came in and did a number of kind of PR photos, and this was one that I came across. And this was actually an even older system that we had developed. But a couple of things I wanted just to point out, the importance of the interdisciplinary collaboration. That was so important in these 11 a.m. tea breaks. I remember occasionally John Leavesley and I would sometimes be dragging ourselves in just about 10:59 because we had to be in there by 11 so that Mrs. Collins, who was making the tea, wouldn't kind of comment about her in a disapproving way, but she was lovely. She was wonderful. But this multidisciplinary collaboration was so important, and I certainly hope that has continued at the IBME up at the Churchill, because that really was important for certainly a lot of people, a lot of the work that I saw. And also Brian's loyalty and his importance to the technicians. I mean, those guys were so important, and Brian really looked after them. He really did. So, and I think one of the key things also that we've all highlighted and will continue to highlight, I think, afterwards, is the importance of using engineering to make a better world. I think that was really important to Brian, and hopefully that legacy continues. Okay, I'll just quickly, very quickly, just talk a little about what I'm doing right now, just because I know we're way behind on time. So this is a company, an Oxford spin-out as well, very pleased to be able to say, that is developing a new kind of test, new kind of blood test to monitor and act in active surveillance for cancer. I don't have the time really to go into great detail about this other than to say that we finished a clinical trial about 2 or 3 months ago in prostate cancer patients, and we're able to show, I'll show you just one quick piece of data in a second, that shows that you can stratify from a simple 10-minute blood test, you can stratify patients in terms of disease severity in prostate cancer patients. This is potentially a phenomenal development, and we've got a number of groups interested and excited about this technology in terms of working with us, and we're working actually setting up clinical trials now in China to look at a number of other cancers, including breast, lung, and colorectal cancer. Again, I won't go into the great detail other than that there's a lot of evidence. What this does in terms of this blood test is it looks at neutrophils, which are a type of white blood cell, and it looks at how they respond to a simple chemical challenge. And this was based on a lot of work that I had done over the years, looking at how a neutrophil is important in a variety of different disease settings. And it was clear that there's a lot of, that neutrophils have a huge impact in cancer. And so that's why we thought, we pushed hard to have this clinical trial started. And I was able to find some great people over at UCL who are very interested in looking at new ways and new biomarkers for different cancers. So this is a simple device that we developed over the years. In a nutshell, you take a tiny bit of blood, you mix it with a variety, some proprietary chemicals that we've identified over many years, and what you're looking for is the white blood cells to respond. If they respond in a certain way, that tells you one thing. If they respond in a different way, it tells you something else. And the response is very simple. It's an optical readout and gives you a quantitative number. And here's just, again, very quickly, some of the data. And you can see, I won't go into great detail, but what you're able to see is that this is all blinded, so we didn't know what category any of these patients were in. A simple 10-minute blood test, we were able to see, you can see the score that we measure from our system on the horizontal axis, and these are the various categories of patients. And after the fact, we were able to see, you can see starting from the less risky, low-risk disease, it's called, still having, these guys have cancer, these probably didn't have cancer. Small amounts, small tumor, slightly larger, and then metastases. You can see a very, very clear correlation between our results and the stratification of these patients. So it's a small test, 70 patients, but it gives us great confidence there's something here and there's something worth pursuing. And again, we think one of the uses of this would be as a home test, and that's really where I see an application in terms of taking this technology and enabling people, especially people who are in remission, to be able to monitor themselves and to be able to take control of some of their cancer monitoring for recurrence. So that's all I really wanted to tell you about the MediStress stuff. I also wanted to tell you just very briefly, again, about some of the work, highlighting the fact that really Brian was all about using engineering to make the world a better place. And so some of the things I've been doing on the side is to mentoring a number of individuals from different countries in Africa for what's called the Africa Prize, which is sponsored by the Royal Academy of Engineering. And I've been very fortunate to actually have over all 3 years of my mentoring, having worked with guys who either won the prize itself. This is Asquar, who's a fantastic guy, developing a really novel water treatment system for Tanzania, a low cost, using some nanomaterials that he's developed. And this is Arthur from Cameroon, who developed, using an iPad to look at cardio responses out in the far reaches of Cameroon, where there aren't hospitals available. And then just laterally, some work with Brian from Uganda, who's developed a jacket to actually look for tuberculosis for far-flung parts of Uganda, where hospitals and medical care is non-existent. So I just want to stand to say, say thank you very much to Brian. I wish he was here, but anyway, thank you guys.